This is Shreya,
And Jackie.
and this is our primary research article on TFG facilitates outer coat assembly on COP2 transport carriers to promote tethering and fusion with ER-Golgi intermediate compartments.
The authors of this research article is Michael G. Hanna IV et al. The majority of the research was held in the Department of Biomolecular Chemistry at the University of Wisconsin, University of Toronto, UC Berkeley, and the Scripps Research Institute.
So starting off with the abstract of the paper, COP2 mediates the initial stages of protein trafficking by putting together subdomains of the endoplasmic reticulum, or the ER, into two layers to basically create carriers that can fuse with the next door endoplasmic reticulum. In the ERGIC, they demonstrated that the TFG bound directly to the inner layer of the COP2 coat. Within that, they found that the TFG C terminus interacted with SEC23 with the outer COP2 coat and the cargo receptor Tango 1 and CTAGE5. In their findings, they found that TFG binding to SEC23 basically outcompetes any other interactions and promotes the outer coat of COP2 to dissociate. They also demonstrated that TFG essentially binds vesicles harboring the inner COP2 coat, which contributes to the clustering between the endoplasmic reticulum and the ERGIC and cells. So overall, the big picture of this study defined a mechanism by which COP2 transport carriers can be retained locally between the interface of the ER and the ERGIC after the outer coat has disassembled, which is necessary in order for the fusion of vesicles with the ERGIC membranes. Just for some quick definitions, COP2 stands for Conserved Coat Protein Complex 2. COP2 is a type of vesicle protein that transports proteins from the rough ER to the Golgi through a process called anterograde transport. COP2 refers to the specific coat protein complex that initiates the budding process. This coat consists of large protein subcomplexes that are made of 4 different protein subunits. The ER is the endoplasmic reticulum, and the ERGIC is the ER-Golgi intermediate compartment. This mediates the traffic between the ER and the Golgi, as well as facilitating cargo sorting. TFG, also short for the Turk fusing gene, this is a protein implicated in multiple neurodegenerative diseases and oncogenesis, and SEC23 is a GTPase activating protein.
The conclusion that was made through this research revolves around the significance of TFG. The TFG C terminus associates direction with SEC23. This association of TFG bound to SEC23 ultimately outcompetes other associations in concentration-dependent ways. It also advances outer coat dissociation, known as uncoating. TFG also restricts diffusion of carriers until the tethering of vesicles harboring the inner COP2 coat, resulting clustering between the ER and ER-GIC within cells. Now I will analyze and discuss Figure 1 of the research article. Overall, Figure 1 reveals that TFG regulates the transport of conventional COP2 carriers. This was done through a series of experiments and immunoblots. Figure 1a shows the immunoblot of various proteins. This was done through different experiments. These proteins were then extracted from vesicles that budded, and the ultimate question was if the protein was in COP2. This showed that the addition of COP2 subunits promoted the formation of transport carriers harboring cargoes. This included ERGIC 53, R-SNARE, and SEC22B. It did not include riboforin, which is an ER-resident protein. Figure 1b shows the amount of procollagen in COP2 transport carriers. The graph shown compares budding reactions performed both with and without TFG. As you can see in the graph, it was found that TFG doesn't help with the formation of COP2 carriers containing procollagen. The image under Figure 1C represents the immunoblots of partially TFG depleted and completely depleted cells. The image on the left shows the 36-hour partially depleted cells, and the image on the right shows the 60-hour fully depleted cells. Actin was also used as a variable that served to quantify the levels. Lastly, Figure 1D shows images of immunostained control cells and TFG depleted cells from the previous image. It showed that Sec24 is always associated with Sec16A. The box on the right, on the bottom, reveals the accumulation of the vesicle bud when you don't have TFG.
So for Figure 1, Part A shows an image of an immunoprecipitation, or an IP. It is a small-scale affinity purification of antigens that uses a specific antibody that's immobilized to a specific solid support, such as magnetic particles or an agarose resin, and it's one of the most widely used methods for isolating proteins and other biomolecules from cells or tissue lysates for subsequent detection by Western blotting or other assay techniques. It was basically an adaptation of a traditional column affinity chromatography, which involves sampling, washing, and other solutions to pass through a column that's packed with resin onto which a target-specific antibody has been immobilized. But instead of using a packed column, IPs use a small amount of resin in a microcentrifuge tube, and incubation steps are performed in a batch-wise manner. For each step, solution is added to the beads, and then they're mixed and incubated together essentially as a mixture. And at the end of each incubation step, the beads are essentially pelleted to the bottom of the tube through centrifugation so that the solution can be removed using a pipette, and you're essentially left with the protein bead at the bottom. So this immunoprecipitation used antibodies directed against the TFG or the TERK fusing gene, and they were conducted using the rat liver cytosol, which was separated by SDS and Page, and then immunoblotted using the indicated antibodies. And as you can see, like the little asterisks highlight the nonspecific bands, recognizing the Sec23 and Sec13 antibodies. For Part D of Figure 2, shows a gel filtration chromatography, or also known as GF, and it separates proteins solely on the basis of molecular size. And separation is achieved by using a porous matrix to allow molecules that have different degrees of access. So, example, smaller molecules have greater access and larger molecules are essentially excluded from the matrix. And for knowledge purposes, SDS Page stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. And essentially what they did is that they used Kumasi staining to allow the bands to show. And the Kumasi dyes, essentially how it works is it binds to the proteins through ionic interactions between the sulfonic acid groups and the positive protein amine groups, as well as through van der Waals attractions. And the solutions of the dye, dark blue black at pH 7, and turn a clear tan upon acidification. And essentially what this does is that this allows, the Kumasi staining allows for the bands to show up as dark blue bands amongst a light background, so it's easier to see which bands are darker, which bands are thicker. So you can see the radius and like the sizes of each band to measure the amount of proteins that are there.
The conclusion reached in this research article is that there is a main mechanism where COP2 transport carriers are retained locally within the interface of ER and ER-GIC. This is vital for the fusion of ER-GIC membranes. Also, it is concluded that the TFG C-terminus interacts specifically with Sec23. Lastly, the clustering between ER and ER-GIC is caused by TFG tethering to the inner COP2 coat. This research is extremely significant because TFG is a protein that implicates neurodegenerative diseases and oncogenesis, which can cause the formation of cancer. So that brings us to the end of our presentation. Here are our citations that we used for our presentation for any references that may be necessary. Thank you for listening.